Hello, everyone. So now what I'm going to do is I'm going to talk about how those sarcomeres contract. And if you're unfamiliar with the word sarcomere, please make sure you've watched my anatomy review lecture that I've already posted. So now what we're going to do is we're going to take those sarcomeres and understand how they contract. Remember, contract means to shorten. So basically what we're looking at is the excitation contraction coupling and the Crossbridge cycle. Okay, so our story is actually going to begin with how it's controlled by the nervous system, and the type of neurons that would control it are called motor neurons. Motor neurons bring information away from the central nervous system. This would be part of the somatic nervous system because it is a voluntary contraction, as in like you think about moving and then it moves. And then all the somatic neurons, motor neurons, would release a neurotransmitter called acetylcholine. A neurotransmitter is a chemical that causes a response in another cell. So it's very similar to a hormone, but instead of being released from a gland, it's released from a neuron instead. So here we have our motor neurons going to the skeletal muscles and releasing acetylcholine. Now I wanted to take this opportunity to introduce another term called motor units. And motor units are different from motor neurons because a motor unit includes the motor neuron, but also all of the muscle fibers that it controls. Okay, so it'll be one motor neuron plus each one of the muscle fibers that it controls, multiple muscle fibers. So if you look at this picture here, we actually have three motor units because we have the red motor neuron and all of the red muscle fibers it controls, and then the blue motor neuron and all the blue muscle fibers it controls, and then all the purple motor neurons and the purple muscle fibers they control. Okay, so three motor units. And that's important because when we get to graded contractions later, you're going to need to know what a motor unit is. So what would be the difference between a motor neuron and a motor unit? Okay, so remember the motor unit includes not just the motor neuron, but also the multiple different muscle fibers that it controls. So into excitation contraction coupling, and this explains how those motor neurons stimulate the muscle fibers to contract. Remember, contract means to shorten. So what I want to do now is take this kind of scary sounding phrase and break it into the pieces. So we'll talk about excitation, then we'll talk about coupling, and then we'll talk about contraction. Let's start with excitation. Here is our motor neuron. The end of a motor neuron is called the axon terminals. And in those axon terminals, you have little vesicles, which are kind of like little bubbles floating in there, and they are filled with the acetylcholine, which is the neurotransmitter. When the electrical signal comes down to the axon terminal, it'll cause those vesicles to fuse with the plasma membrane and release it, just like the exocytosis process that we already talked about before. And that'll release the acetylcholine into this space, which is called the synaptic cleft. That'll then diffuse across and bind to the receptor. Okay, so this process of releasing the acetylcholine, we'll talk about that later when we get into how the neurons work at the synapse. But for now, I need you to know that acetylcholine is released, and then it will bind to a receptor, which is in pink here, that's located on the sarcolemma of the muscle cell. Once the acetylcholine binds to that receptor, that's going to trigger an electrical event, which is called an action potential. And again, we'll talk much more about action potentials later. For now, I just need you to know that it's like an electrical signal, and that once you have an electrical signal in one part of the sarcolemma, that will spread away from it in all directions and along the sarcolemma, And then once it reaches a T tubule, it'll travel into the T tubule. So now we have electricity not just on the surface, but going down into the cell, into the T tubule. Okay, so now it is excited. There's an electrical event happening. We need to take that electrical event and couple it or connect it to the mechanical event of a contraction. And we do that using calcium. So here's how it works. As the action potential is traveling along that T tubule, traveling along the T tubule, it is going to activate some voltage-gated calcium channels. Voltage-gated means that it only opens at a particular charge. So as it travels along the T tubules, it'll open those voltage-gated calcium channels. And because the sarcoplasmic reticulum is really just hoarding a bunch of calcium, just all inside that sarcoplasmic reticulum, when they are open, that causes the calcium to just flood out from high concentration to low concentration. So the calcium will diffuse out into the cytoplasm. So now we have a high concentration of calcium in the cytoplasm. That is going to act as the signal for contraction. Now, so let's talk about how that works. So in purple here, see the purple? This is actin, which is a long protein filament. And embedded on that actin, we have a complex that is called the troponin, tropomyosin complex. So troponin, tropomyosin complex, which is just two protein complexes hooked together. And the tropomyosin in a relaxed fiber, and so in a relaxed muscle cell, so no contraction yet, just relaxing when the calcium levels are low, the tropomyosin fits into a groove along the actin, blocking the myosin binding site. So the myosin cannot get at the actin. And so what happens is so it's blocking the active binding site. And when the calcium is released into the cytoplasm, because of the voltage-gated calcium channel opening, the calcium then binds to the troponin. This piece right here is the troponin. When it binds to the troponin, it causes a conformational change, which just means it changes shape of the tropomyosin and moves the tropomyosin off of the binding site. Now we have an exposed binding site that allows the head of the myosin filament to bind to the actin. That's what we call a cross bridge. Cross bridge refers to myosin heads, which are part of the myosin filament, binding to the actin. And now we're going to go into the Crossbridge cycle, which explains how the sarcomere actually shortens. Okay, so we're going to do the sliding filament theory, where the actin and the myosin slide along each other to shorten the entire sarcomere. Okay, so at this point, what we have is the myosin head binding to the exposed actin site. The physical binding of the head to the actin causes what we call a power stroke. So what I like to envision is if you have a rope hanging down in the middle, the rope just hanging down in the middle, and we're going to climb the rope. The rope would be the actin, and we, our little heads, with our little heads here, we would be the myosin. So once the myosin holds on to the actin, it causes a power stroke, which means it just pulls along the actin, and that pulls the entire myosin structure along the actin. That's the power stroke. Before that can happen, though, you have to hydrolyze ATP, which means you take ATP, take off that last phosphate group, and that cocks it. Okay, so it's a cycle here, so we'll go through it. So we're gonna start up here at the top. At this point, we're at a resting muscle fiber. Okay, resting as in no contraction has happened, calcium is not available, it's still in the sarcoplasmic reticulum. So in this resting in this resting muscle cell, notice that the myosin is not bound to the actin. That's because the tropomyosin is blocking the active binding sites and therefore it can't get at it. And at this point, the myosin head has already been cocked. Okay, so when you think about cocking, think of like spring loaded, like cocking a gun, getting it ready to fire. Okay, so that's going to take some energy, and that energy comes from the ATP. So the ATP has already been hydrolyzed, broken apart. That provided the energy to cock the head. Then from there, because the calcium is now present, the release of the calcium allows the myosin binding site to be exposed, and so the myosin will bind to the actin. When it binds to the actin, that creates the conformational changes that cause the power stroke. Okay, so you hydrolyze ATP, that cocks it, then it binds to the actin, then the power stroke. Hydrolyze ATP, cocks it, binds to the actin, power stroke. Okay, so we have our power stroke here. That's the physical shortening of the sarcomere. And once it does the power stroke, that releases the ADP that's already been spent and used up, allowing the free ATP binding site. The ATP is now going to reattach. It'll hydrolyze it again, cocks it again, and now you can repeat the full cycle. Okay, so please note that ATP hydrolysis is for the spring-loaded, the cocking phase, not the power stroke phase. The power stroke actually comes from the myosin binding to the actin itself. The physical act of the myosin binding to the actin, that's what gives it the power to do the power stroke. The cocking happens because of the ATP hydrolysis. So you cock it, so kind of think about like reaching up for it, binding to the actin, then the power stroke. And it'll just repeat a couple of times, and that pulls the myosin along the actin to shorten the entire sarcomere. So at this point, I think you should pause the video, go over to the PowerPoint slides, and go ahead and watch these videos right here. So pause it, go watch those videos. Then we need to talk about muscle relaxation. So at this point, we've already contracted the muscle. How do you go back to its original length when you relax it? Now, when the important thing to understand for how the relaxation works is that the sarcoplasmic reticulum has active calcium pumps. And the reason that they're active pumps instead of passive pumps is because that means that they're constantly taking the calcium and putting it into the sarcoplasmic reticulum. So they're moving from a low concentration of calcium towards a high concentration of calcium, so that you have a very high concentration of calcium in the sarcoplasmic reticulum. Those pumps work all the time, even during contraction, so that as soon as you release the calcium into the cytoplasm, it'll be returned back into the sarcoplasmic reticulum. And so as you take the calcium and put it back into the sarcoplasmic reticulum, the SR, the calcium levels will return back down to their normal low levels. We now have troponin not bound to calcium, so very low levels of calcium, troponin unbound to calcium. That then changes the tropomyosin to its original shape, and therefore the myosin binding sites on the actin are now going to be blocked so that the myosin cannot bind. And if you cannot bind, you cannot do the Crossbridge cycle, then it'll just passively recoil back to its original length. That's relaxation. So I suggest that you go ahead and pause it and see if you can answer this. Okay, so during relaxation, what's happening to the calcium levels? Is the tropomyosin blocking or not blocking the binding sites, or is the myosin bound or not bound to the actin? Okay, so pause it, see if you know the answer, and then we will continue. Okay, so during relaxation, so when you are not trying to relax, the calcium levels will be low because they are put back into the sarcoplasmic reticulum. The tropomyosin will therefore be blocking the myosin binding sites. And if they're blocking the sites, that means that the myosin cannot bind the actin, and therefore they cannot pull along them. So if we're looking at different types of chemicals, in order to help us make sure we understand the process, let's answer this question right here. If we have two chemicals, one's called chemical A, and chemical A blocks acetylcholine receptor sites. And we have another chemical, chemical B, and chemical B floods the cytoplasm with calcium ions. Of these two choices, which would make a good muscle relaxant? What do we think? I want to pause it, think about it for a sec. The answer would be chemical A, because if you had to have acetylcholine binding, no acetylcholine, no action potentials, no calcium. No calcium means you cannot have contraction because the myosin heads wouldn't be able to get at the binding sites. And chemical B, that would cause a lot of contraction because all those calcium ions would be a signal for contraction. So the answer for best muscle relaxant would be chemical A. So this right here is just for you. This is just a summary. As you go through this, make sure that you really think of it as a cause and effect, right? Don't try to memorize each individual piece. You really need to think about what causes what, causes what, causes what. Like it's a story, not like trivia. Okay, so cause and effect is going to be incredibly important for your understanding. So what I would like you to do right now is go ahead and try and draw this out for me. So what I want you to do is to draw something like this, and it doesn't have to look exactly like this. Just draw something like this. Here is your motor neuron axon terminal. Here is the sarcolemma of the muscle fiber. There's your T tubules. Here's the sarcoplasmic reticulum. There is your sarcomere. Okay, so draw something similar to this. And then on top of that, I want you to number the sequence of events, started with acetylcholine being released from the motor neuron and ending up with the Crossbridge cycle. Okay, so acetylcholine, how the action potentials move across, what happens with the calcium, and then that Crossbridge cycle. So pause it right now and go do that. And then once you come back, I'll show you what I did. So here we have the acetylcholine released. Acetylcholine is going to be released from the vesicles in the axon terminal. Though that acetylcholine will then bind to the receptors, which are shown in green here. That is going to trigger a wave of action potentials that will travel along the sarcolemma and down into the T tubules. Okay, the T tubules, along those T tubules, you have voltage-gated calcium channels. As the electricity, the action potentials travels through them, that'll release the calcium into the cytoplasm. When you release the calcium into the cytoplasm, it's going to bind to the troponin, which changes the tropomyosin, which allows the myosin heads to form cross bridges. And remember, a cross bridge just means you have actin and the myosin connecting to it. That's a cross bridge. The physical act of binding to the actin creates a power stroke, pull on it, that shortens the sarcomere. We then hydrolyze to cock the myosin once again, and then we're just going to continue the cycle through, allow it to pull along the actin. So I have also taken a picture of this, and I will upload it to Canvas as a separate picture on Canvas. Thank you. See you later.